My name is Francesco Del Caratore, and I'm here to tell you about our paper that was recently published in mSystems. In this paper, we present a multi-omics study of Planobispora rosea, the producer of the thiopeptide antibiotic GE2270A. Planobispora rosea is a genetically intractable bacterium which produces GE2270A. This is a thiopeptide used as a precursor for the synthesis of two antibiotics under clinical study for the treatment of Clostridium difficile infections and acne. In our study, we collected a large amount of data both from transcriptomics and metabolomics experiments for P. rosea grown in industrial fermentation conditions. The analysis of this data allowed us to understand the biology of this bacterium. The transcriptomics data showed us in detail how the protein biosynthesis and respiratory chain shut down at the end of the exponential growth phase, when the antibiotics are produced. The data also revealed that both iron and phosphate were limiting the growth of the bacteria in our cultures. This might be exploited to optimize the growth dynamics for the improved production of GE2270A. The transcriptomics data not only showed us that phosphate starvation was actually happening. By analyzing the individual genes involved, we were able to develop the very first detailed description of the phosphate regulon in P. rosea. In addition to the transcriptome data, we measured the metabolomics profiles of the same samples to gain a clearer picture of the changes in the metabolism of P. rosea during fermentation and antibiotic production. We observed the production dynamics of GE2270A and related compounds, and we determined in detail how the level of its amino acid precursors changed, both in the extracellular medium and inside the cells. We also found new metabolites not previously detected in this species. For example, the production of two siderophores was detected by untargeted metabolomics. Examining the rosea's genome, we identified the biosynthetic gene cluster potentially responsible for their production. The data collected in this study were used for the construction and refinement of the very first genome-scale metabolic model for P. rosea. These helped us understand the physiological consequences of the observed gene expression changes. For instance, we could simulate how cell wall teichoic acids are first produced during exponential growth and then replaced with phosphate-free thiocuronic acids during phosphate starvation. This way, teichoic acids could be exploited as phosphate storage by the bacterium. This work was done as part of the H2020-funded Topkapi project, which works on the development of improved chassis for the industrial production of antibiotics. I hope this video kindled your interest and gave you some useful context as you read our paper on mSystems.